and we'll get started. I'm actually the presenter today with my dear friend, Dr. Courtney O'Hagan, and we're going to talk to you about the science of learning. Neurons that fire together, wire together, and I promise that will make sense by the end of this presentation. Courtney, would you like to introduce yourself now or hold on?
Sure. Hello, everyone. My name is Courtney O'Hagan. I am an associate professor and the chair of psychology and human services at SUNY Broome Community College. Yeah, that's all.
And genius too, by the way. She's modest.
She's too kind.
Accurate. All right, so let's move on. I would like to take a moment just to talk to you about what we're going to do in a series this week. We have the science of learning parts 1, 2, and 3. And so today we're going to talk about the brain structures and what's happening within your brain when learning is actually occurring. And then on Wednesday, we're going to talk about the processes and systems of memory, what's happening, what phenomena are happening that help us encode, store, and retrieve that information. And then Friday, we're going to kind of put those two things together, the neuroscience and the learning and memory principles, and talk about some teaching strategies that you can employ in your classroom, as well as some strategies you can share with your students that will help them study better, take notes better, etc. So I hope you'll stick with us as we take you through this journey where we lay some fundamentals down and then get to the application on Friday. So our agenda for today is, first, we're going to talk about some brain myths, what they are and whether or not they're debunked or not. And then we're going to jump into some fun facts about the brain. You might gather by the end of the session that Courtney and I are particularly excited about the brain and all its functions. So we like to share the fun stuff about it as well as kind of like the details about it. We'll pop into some brain basics to give you, like a little bit of foundation before we start talking about learning at the molecular level. And then we'll have a really brief discussion about where memories are actually stored within our brains. So I'm going to hand it over to Courtney, and she's going to handle the next section here.
All right. Yes, we are going to talk about brain myths because there are many that abound. So we can get started with the first one, Jamie. It'll be my slide changer. Okay. So the first myth, perpetuated by movies like Lucy most recently, is that you only use 10% of your brain. But most neurologists would agree that your brain is always active. This is false. We're rapidly firing millions of brain cells. Even while you're sleeping, your brain is active. And maybe this myth came from the idea that not all of your brain is used at once, at every moment. That would be complete overload if it was. But your brain operates on something called the use it or lose it principle. So if there were parts of your brain that were not used, your brain would not put energy and resources into maintaining those parts of your brain. So this is false. Okay. So the second myth is that brain size affects intelligence. And the truth is that size doesn't matter as much as we think it does. I had to go there. Sorry, guys. So this is false as well. Our total brain volume is weakly correlated with intelligence, but not very strongly. And most research found that intelligence is better determined by the number of connections in between brain cells. We're going to call those synapses, and we'll talk more about those later. And also another interesting new thing that they've found, neuroscience has found recently, is that intelligence seems to be correlated with the speed of electrical signals in certain parts of the brain. So higher speeds leads to faster processing rates of information. So fun fact too, just real quick, is that at 12 weeks of pregnancy, this is when we have a huge boom of synapse formation in the brain. So early brain development is important even in utero. Okay, next one. All right, this is a big one that people still I guess, tell students and each other. So there's a myth that left brain people are more analytical and methodical. They're better at engineering and math, while right-brained people are more creative and artistic. This has been debunked multiple times. Scientists at the University of Utah have debunked this myth that people use one side of their brain more than the other, no matter what their sort of skill set is. They examined over 1,000 people's brains and they really found no evidence that you use one side more than the other. So not true. Okay, myth number 4 is that our brain declines as we get older. And the truth is really that this is mostly false. There are some cognitive functions that decline as you get older, but plenty of mental skills improve with age. So there's good news. Things like vocabulary, comprehension, conflict resolution, and emotional regulation all improve with age. So there are some things that older people are actually better at than their younger counterparts. All right. So here's a big one that people often still believe. The myth that people have different learning styles. So this has been debunked in several different ways over time. And I looked interestingly into the history of this. It seems to have become popular in the 1960s and 70s. But really there is no evidence that being taught information in your preferred learning style leads to better learning or memory or even retention of that information. And there are tons of studies that show that this really is not a thing, even though in education, like early childhood education and high school and middle school education, this is still taught to future teachers as a really important way that we can meet students where they are, and we should tailor our lesson plans to student learning styles when actually there was no scientific evidence to show that learning styles are a real thing, that people learn better in a learning style versus another. I would love to. You can actually just Google it. It's pretty simple. It shows up lots of places that this is a myth, but I'm happy to send you some of my resources as well. All right, so myth number 6. I'm sure you guys have seen recently the big push for brain games. There's lots of apps on your phone, but really brain games don't improve learning and memory very much. There was a huge study that was commissioned by the BBC where they made almost 9,000 people play brain games to see if it would improve their reasoning and memory. And they found that even playing these games 10 minutes a day, 3 times a week, didn't really improve their cognitive function in tasks, which they weren't specifically trained for in those games. So this one is a myth too, just a little bit hard to take because we want to try to improve our memory and reasoning skills, but it doesn't help to do that with those games that are marketed that way. Myth number 7, your IQ stays the same throughout your life. Now in psychology, IQ is certainly not a perfect science. There's lots of controversy about how we measure IQ and measure intelligence, but there is some more recent research that's shown that IQ can fluctuate across age, particularly across adolescents. When they look at IQ at 12 versus 16 years old, they see some pretty big leaps in IQ. And intelligence can change across your life, and particularly certain parts of intelligence. So fluid intelligence, which is your ability to think quickly on your feet and recall information, this peaks at about age 18 and then slowly kind of tapers off as you age. But also emotional intelligence is something that can improve across your entire life. So the idea that IQ is exactly the same across the lifespan is not necessarily true. Next one. All right, so this is a fun one that Jamie and I talk about all the time because we are master procrastinators, but we do tend to see that we work better with a deadline. So the myth is your brain works better under pressure, and the answer to this is kind of mixed. The answer is yes to a certain point, but then once you hit a tipping point, there's too much stress and your brain actually does not perform very well. So the pressure of a deadline can motivate you to work harder, but it doesn't always result in better performance. So too much stress can impair brain performance, and you can see that on this graph. This is a really famous concept in psychology called the Yerkes-Dodson law. It empirically demonstrates the relationship between pressure or stress and performance. So if you increase stress slightly, a little bit, you can actually improve focus and motivation on a particular task. And this is when stress becomes what we call good, or eustress, E-U-S-T-R-E-S, eustress. I know it's kind of a weird word. You've probably already heard of it, but so eustress is good stress, stress that motivates people, that can give you a sense of control over what you're doing. But when the stress increases too much, this becomes ineffective, where the stress is actually impairing your ability to learn. So interestingly, studying, that sort of transient stress of studying, can be considered eustress if it's done at the right time. So that can promote some conditions for optimal learning. So studying right before, 5 minutes before, which sometimes I walk into my classroom, I see my students studying right before the exam, that's probably not eustress. But studying, a couple days before the exam, that can be motivating enough to actually increase learning, be considered eustress. So eustress results in release of hormones like adrenaline and cortisol. And in small amounts, these can actually enhance our connections between our neurons. But in large doses, these stress hormones can impair learning. So we have to kind of figure out where that cutoff point is for ourselves. And then also, it might be nice to teach our students this too, because maybe that's something they can incorporate into their studying habits, is when is a good time to study, when it's a little bit motivating, but not too stressful. Okay. Myth number 9. Human brains are excellent at multitasking. We are, in fact, pretty terrible at multitasking. And it's funny that we have sort of encouraged in our society this idea that we all should be multitasking, because human brains really are not built to do 2 things at once. We cannot perform 2 tasks that require high-level brain functioning at the same time. And the thing is, we often think we are multitasking. We think we're fairly good at it. But actually what we're doing when we're multitasking is switching our attention very quickly between tasks. So think about talking on the phone and writing an email at the same time. It's impossible to do both, but you can sort of switch your attention between 2 things and sort of do both kind of well, but not very well. So when you switch from task to task that quickly, you're not actually really paying attention to everything at the same time, even though you think you might be. And we see the evidence for this in fMRI studies, where we look at people whose brains are trying to complete competing tasks at the same time. And we see that the attention switches and the parts of the brain that you're using switches, but you're not actually doing both at the same time. Okay. And then I think it is brain fun facts with Jamie.
Brain fun facts. All right. So we're going to switch over. I was just having some trouble multitasking with typing in the chat and moving our slides along, so we can see already that multitasking is less effective. So I'm going to pop us into some fun brain facts that I like to share with my students in particular, because it blows their minds and it's kind of fun to talk about. So first, my favorite image of the brain is our brain is about 2% of our total weight, but it consumes 20% of our energy. So for something that is relatively small, it's consuming a vast amount of energy, because as Courtney said, it's constantly working, even while we're sleeping. Our brain is electric. It actually communicates both chemically, but also electrically. There are actual electrical signals that are generated within our brains. And while awake, our brain actually generates enough electricity to power a light bulb. Only 10 to 23 watts of power, so like a dim light bulb, but a light bulb nonetheless. So I hope nobody is, like squeamish, but this is actually an image of a brain that has had the blood vessels perfused. And so they kind of stay in shape when the rest of the brain tissue is removed. But if we were to lay out all of these blood vessels in a line, it would span about 100,000 miles, which is 4 times around the globe. So we have a lot of blood vessels in there that are kind of keeping our neurons and other supporting cells nourished. 60% of our brain is composed of fat, and not the kind of fat that fills this cat's belly. It's a special kind of fat called myelin, which our brain needs in order to insulate parts of the neurons that are conducting the electrical transmission, the electrical communication. It works like the kind of insulation around the cord that goes from your lamp to the outlet, so that the electricity doesn't, shock anyone or kind of go where it's not supposed to go. That fat in our brain acts as that insulator. So, if someone says you have a fat head, you're like, great, that helps me. That's my insulation right there. Signals within the brain can travel at 268 miles an hour. And for reference, the Bugatti is the fastest car on Earth, and that travels at 268 miles an hour. So the driver of the Bugatti is actually traveling at the speed of thought. For reference, high-speed trains travel at about 374 miles per hour. So they're traveling much faster than the speed of thought. And but nonetheless, that fast transmission between cells is what makes it possible for us to process so quickly, for us to react quickly to like something hot on the stove, or to present like this, right? So this is one of my favorite quick little videos to show to my students in class, and it kind of references that the brain itself has 0 pain receptors. The pain receptors are located on your scalp, at the surface level. So when surgeons are actually conducting neurosurgery, they only have to anesthetize the outer layers of the skull. They don't have to worry about being inside the brain. So once they have that hole open, they can actually have patients awake during surgery, because they can poke around there very carefully and make sure that they're in the right location. So I'm going to play this video.
Man, the surgeon in me is antsy. I need to cut somebody up. Thanks for volunteering. I'm not really that strong on the brain. I think this is the language center.
Really? Because I don't feel it. How cool is that?
And this is the foreign language center.
Impossible! Je n'ai pas parlé.
The cow says. The sheep says. Ladies and gentlemen, Macaulay Culkin.
So that kind of talks about how when a surgeon is in there, they can be asking the patient questions and the patient can respond. There was actually a violinist who was having surgery and was so concerned about being able to continue to play the violin that she was actually playing the violin during the surgery while the surgeon was speaking to her. While the brain doesn't have pain receptors, we do get things like migraines and headaches, but that's primarily associated with the stretch receptors in all of those blood vessels we showed you a couple slides ago. All right, so most people dream for an average of 1.5 hours per night and have about 4 to 7 dreams per night. We have like a 90-minute sleep cycle that we go in and out of throughout the night, and that kind of shifts. We spend more time in certain sleep stages at the beginning of the sleep than we do at the end, but that's where we're cycling in and out of dreams throughout the entire night. Sleep serves a really important function in the consolidation of memories, and we're going to talk about that in more in depth, but we should keep in mind how important that sleep is, because that's where we're actually doing some processing about what's happened during our day, what we've learned, and it's kind of getting gelled in there, solidified while we sleep. During dreams, your brain releases a hormone that prevents bodily movement. And this is kind of critical, because you can imagine in dreams you're doing a lot of moving. You might be doing some sort of activity like Ron Swanson in Parks and Rec, where he sleep fights, right? So if you were lacking that hormone, or you weren't paralyzed while you were sleeping, you'd be fighting while you're sleeping, and that might be harmful to somebody or yourself. The comedian Mike Birbiglia actually has a sleep disorder where he acts out his dreams. He has to be kind of mummified at night so that he can't harm himself or others while he's sleeping. He talks about that a lot in his comedy sketches, and it's hilarious. So you should absolutely give that a listen. Laughing at a joke requires activation in 5 different brain areas. It's humor is a very complex function, and we're not sure if it happens at animals that are lower on the evolutionary scale, at least not that we understand their humor maybe. But laughing at a joke for us involves activation in multiple areas, and it shows you here in these scans how much has to be active in order for us to get a joke. The average human is estimated to have over 70,000 thoughts a day. And that is just mind-blowing to me to think about how many separate thoughts we're having. And some of them are happening kind of below the surface, those implicit thoughts, perhaps like we're maintaining our balance, or we're thinking about, how shifting in your seat, makes you feel. But on average, we're thinking quite a few thoughts a day. STM here is abbreviated for short-term memory. So our short-term memory can hold on average a maximum of 7 digits. So back when we used to have to memorize phone numbers, right? When we used to have to use, our friend's phone to call our parents, or we would have to call from the payphone home to have them come pick us up, we would have to memorize phone numbers. We don't have to do that so much now, but that kind of maximum that our short-term memory or working memory can hold is about 7 digits. And that's the same as a telephone number. We'll talk about different ways to expand that short-term memory tomorrow when we talk about memory processes and systems. And finally, for brain facts, learning something new actually causes a physical structural change in your brain. And this is the amazing thing about what we call our plastic brain. That plastic, in terms of neuroscience, means we have the ability to change it across our entire lifespan. It's not just, when we're growing in utero in those first 2 years post delivery. It's throughout our whole lifespan that our brains are growing, they're pruning things out, et cetera. So these learning new things are causing new connections, new synapses to grow, et cetera. So we're going to switch on over to some brain basics, and that's going to be Courtney's section here.
Okay, so we're going to give you a slight crash course in neuroscience. Remember, this is not a biology class, so we're going to talk about it in a very basic way. This is how I teach it in intro psych too. So my students get a really basic view of a central nervous system and how neurons work. So our central nervous system is the system of our body that's really specialized for communicating between the brain and the body. And the parts of the central nervous system are the brain and spinal cord, which you can see in that picture with the red background. That is what it would look like if we took your brain and spinal cord out of your body. Not at all gross, but kind of cool. So I said this already, but a main job of our central nervous system is to communicate. And your central nervous system, particularly your brain, consists of neurons, or sometimes called nerve cells. And there are a few different kinds of neurons, but in a minute, we're going to talk about the structure and function of neurons. But there are specialized cells for communicating using both electrical signals and chemical signals. So your neurons use electrical signals within the cell, and they use chemical signals between 2 cells. So we consider your brain to be the processing center of your nervous system because it integrates all of the incoming information from your body and then processes what you're experiencing and sends messages back out to the body. Your brain consists of around 86 billion neurons. And that's really difficult to comprehend. And that's why I think Jamie put this great picture over here of money, because all of us can kind of comprehend the size of a dollar bill or $100 bill. So yeah, you can't even really comprehend how much a trillion is, but 86 billion is very close to a trillion. So that's the concept we were trying to convey there. Okay, we can go on to the next one. We're going to compare brain size next. So this is a comparison of brain size between lots of different animals, mammals. And if you notice, the human brain down on the right is a much bigger. But also the main thing to kind of take away is that it's much more wrinkly looking. And those wrinkles, most of you probably know, but those wrinkles are on the cortex of the brain and they are called convolutions. So the more convolutions in the brain, the cortex, sorry, a species has, the more intelligent it is considered to be. Although there's a little controversy about that as well. But in general, the more convolutions the brain has, the more cortical tissue it can kind of squeeze into the size of your skull. Okay, we'll go on to the next one now.
Multitasking, I'm sorry, I was chatting.
All right, so this one we'll spend a little time on so Jamie can answer some questions. So here's the thing, your brain takes decades to mature. And I'm sure any of you who have children completely understand this and will get it. But there's a lot of things I think we can take away from this that might help us in our teaching. So our brain doesn't reach its maximum size until you're about 18. And even then, it's still not done maturing. So something to consider is that as our brain is developing throughout childhood, that circuitry, those neural connections that originally are adapted for basic survival functions when you are an infant, like recognizing our parents' faces, those circuits have to reconfigure multiple times as we learn new information. So we have to take complicated topics that we have a kind of structure for as children, and then feed into it the new complicated information that we learn across the lifespan. So across childhood, when the first few things that we learn to do that are much more complicated than basic survival skills are things like reading and math. And our brain has to figure out how to scaffold this new information into existing neural circuits to expand those circuits and strengthen the connection. So all the information we learn requires a basic structure in order for us to fill in that information as we learn. And as Jamie says, you can't teach a baby calculus, which always sounds funny and obvious, but you start with basic things, right? You teach children things like numbers and symbols, and then you teach them addition and multiplication and algebra, and then eventually you get to pre-calculus and calculus. And that has to do with feeding information into the existing structures of our brain to connect, to expand those circuits and those connections. And this ability of our brain to really be changeable and to reconfigure connections is referred to as brain plasticity. So you've probably heard that word before. Plasticity is much greater earlier in development, but that doesn't mean that we do not have plasticity across the course of our lifespan. Our brain does continue to be plastic throughout adulthood, but it is a lot more plastic during childhood and adolescence. So I think what I wanted to add here really is the most important thing for, I think, educators of college students to understand about brain development is that our brains really are not fully developed until we are about 25 to 26 years old, which is even past college age. So we are teaching students whose brains are not fully developed. And there's a few key structures that really are not fully mature until this time. Most importantly, is this part of our brain right in the front called the prefrontal cortex. This is where reasoning, strategizing, and decision-making is happening. And this is not completely developed until people are about 25 or 26 years old. So if we think about how to apply that information to how we teach students, some scientists have suggested that our assumption that students have self-discipline and the self-discipline to study regularly for far off deadlines, like to plan ahead really, weeks and months. And we have this expectation that students know how to sequence very large projects and integrate them, that those ideas that educators have and expectations that we have may be a little bit misguided, because that part of your brain that would allow you to plan ahead and kind of calculate how much time you have left before a project or before an exam, that part of our brain isn't fully developed until we are past college age. So I'm not saying that college students can't do those things, but I think it's good to take into consideration that these are not skills that they are completely adept at yet. However, there's a lot of recent neuroscience that shows that college age brains are in sort of a golden period for learning, which is excellent news for us. They still have more ability to learn than adults, and their brain cells are more active and more receptive than adults and can be shaped more readily by experience than the later adult years. So it's kind of good and bad news as far as brain development when we talk about college students. Okay, we can go on to the next one, Jamie. So I talked a little bit about this before. This is a picture of the wrinkly part of our brain. I always show students sort of as your brain develops, looks kind of like a fist. And the last part to develop and grow in utero is the cortex, and it grows over the top. The cortex is sometimes called the cerebral cortex or the neocortex. So if you hear those words later on, you'll know we're talking about the same thing, the same structure. It's a giant structure. And so we have to break it up into many functional parts in order to talk about it more fully. But the cortex is really where all the magic happens, the magic of being human. This is where our higher level cognitive processing happens, thinking, planning, analyzing, knowing, processing the difference between the sound of a dog barking and the sound of your mother talking on the phone. Not that they sound anything alike, but the fact that we can recognize that difference has to do with what's happening in the cortex. So I'm going to talk to you a little bit about the parts of the cortex before we move on. We have several lobes. So the cortex, in order to talk about its functionality, is divided into 4 lobes. You've probably heard of these lobes. We have the frontal lobe or the frontal cortex, which spans the front part of your brain. I guess Jamie is doing the cursor, but I of course do it, with my hands. I'm not even Italian, but I do it with my hands. Because it helps students, I think, in the class understand. Anyway, so your frontal lobe contains parts of your cortex like the motor cortex, which runs on both sides. This is involved in processing complex movements and producing movement in the body. Then the important part I talked about before is that prefrontal cortex right above your eyes and between your eyes. That prefrontal cortex is involved in things like decision-making, planning, self-control, emotional regulation. It's involved in so many important things. The next lobe is the parietal lobe. So your parietal lobe is kind of in the very center, top of your head. It runs down both sides. And this parietal lobe contains a part of the cortex called the somatosensory cortex, which processes touch information, pain, and temperature information from the body. And then we also have the temporal lobe, which runs on both sides of the cortex, kind of right above your ears. So your temporal lobe is the part of your brain that it does a couple other things, but most importantly, it contains the auditory cortex, which is the part of our brain that processes sound information and recognizes language, things like that. It also helps produce language. And then in the very back of your head, that part is the occipital lobe, and that lobe contains the visual cortex. This is where you process everything visual. So if you close your eyes for a minute, you're actually not activating your visual cortex, but if you try to picture something inside your head, like the McDonald's sign, you will activate the visual cortex because that is where a lot of imagery is stored. And actually, when you're dreaming, that visual cortex is very active as well. So it processes visual information. Okay, we can move on. So here are the hemispheres of the brain. The brain is divided into 2 halves. It is a mirror image of itself on either side. We call that in biology bilateral organization. So bilateral symmetry or bilateral organization means that the right and the left side look exactly like a mirror image of each other. And in the cortex, they are a mirror image, but one side is a little more specialized than the other. So the right is more specialized for things like spatial relationships and understanding creativity and artistic and emotional sides of parts of our functioning. And then the left hemisphere is really more specialized for motor control. It's more specialized for speech and language and calculating numbers, time, and sequencing, that kind of stuff. But what's interesting is the left side of the brain control motor wise or movement wise controls the right side of the body, and the right side of the brain controls the left side of the body. And there's a crossover that happens. And I looked at a really interesting video that talked about why that maybe happens. It has to do with survival. If you had a predator come up from the right side of your body, your best strategy would be to move to the left side. And maybe that's why there's this crossover of information based on how we process things like visual information. Okay, we can move on from here. All right, so here's our crash course in the neuron itself. So these are our brain cells. We have 86 billion of these guys in our brain. And this is a kind of prototypical look of a neuron. They don't all look like this, but they do all contain the same structures because the structures in the neuron are what help it with the way it functions. So up here at the top is the soma, or the cell body, and off of the cell body, like, looks like a big lump right there in the middle. And off of the cell body are these branches, which are called dendrites. We'll talk more about those in a minute. And then there's a big kind of cord that comes down off of the cell body. We call that the axon. We'll talk more about what that does in a minute. And there are little lumps, almost like beads on a necklace, across the axon. That is the myelin sheath that Jamie talked about earlier, that is made of fat cells, and it helps insulate the electrical movement of the electrical signal down the axon, because that's where I said there's electrical communication within the neuron. That's what's happening in the axon. And then at the bottom of the axon, it can branch even further into separate axon terminals. So there are multiple axon terminals at the end of the axon. And that is where neurotransmitters are stored until they're used, they're released from the axon terminal. And at first you might say, that's a lot of information, that doesn't really make sense. But then once you put it together of how neurons communicate with each other, it makes a little more sense. So we're going to talk next about dendrites, because we're going to kind of zoom in on some of the parts of the neuron that are helpful, particularly when we talk about how learning changes the brain, because Jamie did mention that learning physically changes the brain. Jamie, how am I doing on time? Should I go a little faster?
It's 10:37, so I think we're pretty close.
Okay. So dendrites, remember I said, are the branches off of the cell body, and the more dendrites a neuron has, the more ability it has to connect to or talk to neighboring neurons. And when we learn, this is one of the structures of the neuron that changes. We get branching and growing of dendrites so that neurons can become more connected to other neurons. The thing about dendrites that's also important to know is that they have little receptors. I guess they're microscopic for sure. And the receptors are even more microscopic. On the length of the dendrites, there are receptors for neurotransmitters, and that's what tells that neuron that it needs to become activated or not. So dendrites are how neurons talk to each other. Okay, we can move on. All right, and then remember I mentioned the axon was the long cord stretching away from the cell body. This is where the electrical signal passes to basically activate neurotransmitters that are stored in the axon terminal. So when a neuron becomes activated, like you have a thought or you want to move your pinky finger, and this is a very basic way of explaining it, but what happens is a neurotransmitter binds to, and I love that I'm trying to point to my screen because I'm used to teaching this in the classroom, but a neurotransmitter binds to a receptor on a dendrite. Then the cell body decides whether it needs to pass, decides is kind of a broad term, but whether it needs to pass that electrical signal on and pass the communication forward. So an electrical signal is generated right at the base of the cell body if the neuron is going to become activated. And that electrical signal shoots down the axon faster if there is a myelin sheath, and it shoots down to the axon terminal, and then the axon terminal releases neurotransmitters into another thing we're going to talk about in a second, which is called the synaptic cleft. So those neurotransmitters are released from the axon terminal into a tiny microscopic space between two neurons. We call that, those are those synaptic connections we were talking about. It's this the tiny space or connection between one neuron and another. And in general, it's the axon terminal of the first neuron and the dendrite of a second neuron, and that's where the communication is happening. So neurotransmitters are released from the axon terminal of the first neuron. They float across this tiny microscopic space that we didn't even know existed until we invented electron microscopes. We thought all neurons were kind of touching each other, but they're not. Neurotransmitters float across that space and they bind to receptors on the dendrites of a neighboring neuron. And then that neuron has to figure out whether it needs to pass the signal forward or not. And I say figure out because unless we get into the neurochemistry of it, which we don't have time for, that is a more basic way of talking about it. So the axon varies in length. Some axons and some neurons are short, some are really long, some have multiple axon terminals, and really the job of the neuron is to find other neurons to communicate with. And as we learn new information, that's what changes. Our actual neurons change and become more connected and interrelated to each other so that we form these patterns and these circuits that help us complete tasks with more proficiency later on as we strengthen those connections. Other slide, I'll knock some things over, and then it'll be Jamie's turn.
So now might be a good time for us to pause and see if anybody has any questions. Like we've bombarded you, like the myths and facts were one thing, but diving into kind of like the actual structures of the brain or in like the neuron itself. Does anybody have any questions about that? Are you all right? We're not expecting you to memorize all these parts, right, in order to understand on Friday, but it's kind of helpful to understand these basic component parts of your brain in order to understand these later sections. So is everybody all right? So when a circuit is strengthened, what is physics? We're getting right into that. That's a great question. That's what we're going to talk about learning at a molecular level as we go through the next couple of slides. So great question, and we're going to get to it. Okay, thanks you guys. We just want to make sure that we've still got you guys. This is a lot. We get really excited about this and we forget that not everybody has as much maybe enthusiasm for the brain as we do. So, oh, good. I'm so happy to hear that. So we get really excited. So then let's move on to what's happening there as Courtney was alluding to at the molecular level between the like presynaptic cell, which is like we'll call cell A, and the postsynaptic cell, which is cell B, right? The one sending the neurochemical message and the one receiving it. We're going to talk about this in a little bit more depth, but first we're going to talk about a really interesting person who, an unfortunate series of events actually changed neuroscience forever. It changed how we viewed neuroscience and learning and memory in specific. And so this was somebody we learned about, in our psychology textbooks and in graduate school. We referred to this person as HM because at the time when I was in undergrad and in grad school, he was still living. And I think, Courtney, he passed away while you were in grad school, correct? And so it wasn't until after his death that we were able to know who this person was. So we refer to him as HM. And his name is actually Henry Malayasan, but I'm going to continue to call him HM because that's how I've connected that information in my brain, and it's easier for me to retrieve it as HM. So when he was a child, he suffered a brain injury that resulted in some fairly significant seizures that continued to worsen as he aged. And in 1953, as a young adult, he actually underwent what was called a psychosurgical procedure, but what we now refer to as an experimental procedure that was not conducted after his surgery again. So what they did was they actually removed sections of his medial temporal lobes, which included a very significant structure, which we'll come to find later in this presentation, called the hippocampus, as well as parts of his amygdala. And you can see here this image of a normal brain from the underside. So this is the back of your brain here. This is the front of your brain, your prefrontal cortex. And this is what it would look like if someone was looking down at somebody standing on their head, right? And so this is a normal brain. And with HM here, they've removed these sections of his brain on both sides. So remember, when I'm talking about a structure, as Courtney referred earlier, we have bilateral organization of our brain, or bilateral symmetry. And so if we have one hippocampus on one side, we actually end up having two hippocampi. So they removed it on both sides. And so if you were to look at someone and they were to have the front half of their face like opened up like a book, this is kind of the view we're looking at now. So in a normal brain, you would see the hippocampus here. In HM's brain afterwards, you can see this deficit here on each side where the hippocampi were removed. So what they found immediately post-surgery was that the seizures stopped, but Henry had lost the ability to form new conscious memories. If someone would walk into the room and they left, and then they came back in a moment later, he would have no memory of having ever met that person before. If he had breakfast and they removed the breakfast tray from his room and they came back in, and he might say, "I'm hungry, I need breakfast," he would have no memory of having eaten breakfast. He was unable from that point, and like at the age of 27 all the way up until 2008 when he passed away, he was not able to form these new conscious memories. What we did find with HM was something really interesting, and we'll talk more about this when we talk about the learning and memory processes. But he was able to form these new implicit memories. So like these so-called muscle memories, we talk about that a lot. When you've learned something so well, it becomes this procedural muscle memory. You don't even have to think about it. Driving is an example. Playing an instrument is an example. So he was actually able to develop these new implicit memories even without the use of his hippocampus, which is interesting and comes into play when we talk about the different types of memory. And we'll talk about the structures that are involved with those two different types. Like there's the factual information and there's this procedural information. And we seem to have two different brain structures that control each. So he was able to learn how to ride a bike. He was able to learn how to play piano. He was able to do these tasks where they sat him in front of a mirror and he was supposed to trace the outline of a star by looking only at the mirror. And he was able to show improvement over time, even though if you were to ask him, he had no intellectual understanding of how he did that. So he was studied by neuroscientists for more than 50 years until his death in 2008. And upon his death, he actually his brain was donated to science, and the brain itself was fixed in formalin and then was sliced with a microtome, which allows us to cut tissue into really, really thin slices, like 1 cell deep kind of thing, so that we were able to take a really close look at his brain after he had passed away. And they actually live streamed that. It was one of the first live stream events. And I don't know, Courtney, if you remember where you were when that happened.
Oh yeah, I was in the lab watching it. But yeah, it was one of the first live streamed anything. It's interesting.
Yeah, I was actually at work that day, and I have to say I didn't do much work. I mostly just watched that on my screen, and it was just amazing to watch this brain that I had learned about for 20 years, being maybe 15 years. That was a while ago. So like 15 years being actually dissected in front of us. It was amazing. And so the researcher that worked with him the longest, her name was Dr. Corkin, and she wrote a book called The Permanent Present Tense about him, which I think is a clever title. He was always in the present. And it's a kind of retrospective of his life, a nice look at who he was as a person, who he continued to be, even with this major deficit. And she writes in there what I think is an amazing quote. And he said to her, "It's a funny thing. You just live and learn. I'm living and you're learning. And so this horrible thing that happened to this man actually helped us move our understanding of learning and memory decades forward. It was amazing. So I'm really grateful to have this as a part of our presentation today. And we'll be referring back to his case because it's so incredible to talk about throughout our next two presentations. So let's talk some more about that synaptic plasticity that we've talked about. We talked about it, in the brain facts. We talked about it again with Courtney's when she was talking to you about the actual synapse and the synaptic cleft there. So our brain is able to form these memories and rewire itself completely in response to experience because these circuits in our brain change at these synapses. So this ability to remodel themselves is fundamental. And like Courtney said, we have these discrete periods in our development where synaptic plasticity is at its peak. We have certain critical windows like language development, from 2 until 5, that's the best time really to learn your new language as well as a foreign language, et cetera. So that's when our synapses are most plastic. Encoding a new long-term memory, it involves these persistent changes in the number and the shape of the synapses themselves, as well as the amount of neurotransmitter that gets released from that cell A, that presynaptic neuron, and the number of receptors that are actually located on the dendrites of the postsynaptic cell. I'm going to explain this in more detail in the next couple slides, so don't panic if it just seems overwhelming. We'll get there. First, I want to talk about one of my favorite nerds. This is Dr. Eric Kandel, and he's actually a Nobel Prize winning neuroscientist for his work that moved forward our understanding of the molecular level of learning. And so we have learned a lot about the ways that the presynaptic and the postsynaptic neurons remodel themselves. And this sea slug here on the left is called the Aplysia californicus. And it was an important animal model for Dr. Kandel here to understand synaptic plasticity. Because the sea slug's nerve cells are relatively few, they're really easy to observe. So researchers were able to identify the chemical and the structural changes in the relevant nerve cells of this sea slug that correlated with simple forms of learning and memory. So as the sea slug learned, you could see these conformational changes in the synapse happening. So that was what moved us forward from the kind of global look at learning to this really micro look at learning, and really change things long term. So that brings us to the title of the talk today, which is put forward by Heb, that says, and I'm going to read this because it's important that I say it exactly. And it might not make sense to you, but that's okay. We're going to break it down. Any two cells or systems of cells that are repeatedly active at the same time will tend to become associated so that activity in one facilitates activity in the other. All that means is cells that fire together wire together. So cell A fires and it activates cell B, and then cell A fires again, activates cell B. And the more often that happens, the more connected, not physically, but the more, for the most part, more connected they become, and the greater our learning. That's what this Hebbian concept of cells that fire together, wire together means. Your memory becomes more secure because more and more synapses have processed that information with a repeated exposure, and their connection has become strengthened. Ooh, we're running out of time. I talked too long about HM, I get excited. So we'll talk about this on Friday, but there's an author called Dr. Oakley, and she actually presented at CIT a couple years ago, but she talks about the Hebbian concept in terms of learn it, link it. And this is what happens. So say we're learning our times table. We have all of these neurons here that are kind of loosely arranged together. And we start saying 1 times 5 is 5, 2 times 5 is 10, 3 times 5 is 15, right? And as we do that, cell A is talking to cell B is talking to cell C, and we're going to start to see these connections becoming more solidified. And that's what we see here in picture B. The cells, you definitely see a series of cells. You're learning those times tables, you're memorizing it, you've been working on it for like a week. And then what we see in diagram C is you've been practicing this for a month and you know it down pat. You can say it in the shower, you can say it while you're riding your bike, you can teach it to somebody else. And that's because we have all of these brand new connections made together. And the challenge is for us to take our students from this learn it phase to the link it phase to the really know it phase, right? Is this why it's hard to unlearn incorrect? Yes, absolutely. That is a great observation. It is absolutely essential, which is one of the techniques, right? So when you're working with students, don't send them home where they can learn it the improper way. You know, you work with them in the classroom so you can show them the right way, and then they practice it the right way and not the wrong way. All right, so let's get to the science. Oh, wow, we're running out of time. I'm so sorry. So thanks in part to HM, scientists now know which parts of the brain seem to be involved in kind of conscious learning. And the three main parts that we're going to talk to are the neocortex, which Courtney described as going around the outer edge of the cortex, around all those convolutions, right? And oh, sorry. Sorry, I've lost myself. I've frazzled myself. So anyway, I like to think about the neocortex as a newspaper, right? It's about the thickness of a dinner napkin and 24 by 24 inches square. And it's kind of crumpled up like a newspaper to fit into the brain. And that's where the majority of our long-term memory seems to be stored. Then we have the basal ganglia, and these are implicated in procedural memory. So if you remember from HM, he was still able to form new implicit memories, right? And that's because his basal ganglia were intact. The basal ganglia are associated with kind of this executive function of motor movement, and it's involved in procedural memory itself. And we're going to learn more about procedural memory tomorrow. You're welcome. I try to make my presentations as inclusive as I possibly can. So this is a kind of an artistic rendering of the hippocampus. And this is actually hanging in my living room, if it gives you an idea of how much I love it. It's done by Greg Dunn, who's a neuroscientist. And the hippocampus is really important for the type of memory we're going to be talking about with our students. And that's that semantic, factual information learning. And it has each hippocampus, because we have two, one on each side, right, is about the size of two big lima beans. So if you know how big a lima bean, that's two of those on either side of our head. And it's involved in that declarative learning. It works sort of as an index or a directory. It doesn't store the memory itself. It actually kind of directs where that memory gets stored in that neocortex. So there's this loop of information that comes from that neocortex, that outer layer of our brain, into that hippocampus that kind of helps us learn. So as we're making these connections, we're seeing this kind of transition from our working memory there in the hippocampus to long-term memory in our neocortex. And it's here where we see that molecular cascade that we described in the Heb cell, right? That is essential for those memories to become long-term. The more we use it, the more those synapses fire together, et cetera, the more kind of consolidated that memory is going to be into the long-term. All right, and then for the final slide here, but the question is, where are these memories stored? And there are neuroscientists who argue about, well, it's stored in the synapses. Well, it's stored in, actually, there's a genetic modification. There's a genetic alteration in the neurons themselves. That's where the memory is stored. But for our purposes today, we're going to say that memory is stored in the neocortex. It's in various areas throughout the neocortex, depending, on whether it's a sensory memory, whether it's, factual information, et cetera. And sometimes it's stored in multiple places, right? And sometimes we have to go to both places to pull that information. And you saw that with the humor slide, right? But for the most part, that's how it works. Each part of the brain most likely contributes differently to permanent memory storage. So that was a lot of information that we threw at you today. And we're going to say it's to be continued. Tomorrow, we're going to talk about learning and memory, the systems and processes. So we can stick around if anybody has any questions. But for now, there's a 15-minute break before our next presentation at 11:15. So thank you to Courtney for doing this with me today. I appreciate it so much.
You're welcome. Thank you, everyone, for your attention.
Yeah, truly. I hope you were as excited about the brain as us. So if any questions, please let us know. I'm going to pause the recording so we can move forward to the next presentation. Thanks, everybody.